Okay, so thank you so much, Sarah, for having us, and thank you everyone for joining. My name's Rosie, and this is my colleague Szi Kay. And as Sarah said, we're from the Complex Disease Epigenomics group down at the University of Exeter. And today we're going to describe to you some of our long-read transcriptome sequencing data. The majority of it is unpublished, and we're hoping to make all the data open access when the time comes. However, we're really keen to collaborate, and if you are interested in anything, please do get in touch. Our email addresses are here at the bottom, and they're also on the last slide as well. So what we're going to describe to you today is part of a larger set of projects at the University of Exeter. And we're interested in the regulation of, the gene regulation across the life course, and the role it plays in neurodevelopmental disorders and in neuropsychiatric disease. So there are many levels to gene regulation, going from the genome to the epigenome to the transcriptome, all the way down to the phenome. We're interested in how the mechanisms occur during gene regulation and how this occurs in development and in neuropsychiatric disease. Today, we're going to be mainly focusing on the transcriptome, and we're not only going to be discussing isoform abundance, but also alternative splicing events and how they caused diversity. With over 20,000 genes, with adding in alternative splicing, that adds a whole other level of complexity.
Alternative splicing, as many of you may know, is a very important post-transcriptional mechanism that's involved in particularly in the CNS, where it impacts on function and development. So, for example, here you see a gene with the exons and introns. So exons are protein coding, which is the dark blue, and introns are non-coding regions. And this basically, through alternative splicing, generates multiple mRNA isoforms of different combinations of exons. And these mRNA isoforms can have potentially very different functional effects. And historically, alternative splicing or kind of transcriptome profiling was performed using Illumina short-read RNA sequencing, where basically on a flow cell, through sequence by synthesis, wash and scan, you generate these very short 250 base pair reads. However, more recently, in the last 5 to 10 years, 2 long-read sequencing platforms have emerged. So that's the PacBio, Pacific Biosciences Isoform sequencing, where you basically sequence read multiple times through single molecule real-time sequencing, and the Oxford Nanopore platform, where essentially you sequence the read through a pool, generating kind of this current, where you can therefore by proxy read your sequence. Both these platforms generate very long reads over 10 kb. And basically with this generation of these very long reads, it improves your annotation of these transcripts. So for example, if you go back to a gene of interest, gene N, you have 2 transcripts which are generated through alternative splicing. If you have very short reads, it becomes very difficult to align to your transcript of interest, but also to know what transcripts are present because they're so short. However, with long reads, you basically resolve the need for this transcript assembly. So through long reads, you can sequence your transcript from the 5' to the 3' end, and essentially you can now very quickly and effectively know which transcripts are present.
I'm going to give a brief overview of our lab methodology. So we have 2 data sets. We've got human cortex and an Alzheimer's disease model, mouse model. And we have 2 approaches to our long-read sequencing. We can either perform whole transcriptome or targeted. And I'll discuss in more detail the targeted approach later. But both these approaches involve extracting RNA from our tissue doing cDNA synthesis. And at this point, you add a barcode so that you can uniquely identify your sample. You then do large-scale amplification on the cDNA and do an equimolar pool of samples. Now, at this point, you might do the target capture, which is for the targeted long-range sequencing approach only, and that's where you're enriching your cDNA. But otherwise, you would just proceed with your cDNA to library preparation. And we can either do Iso-Seq, which is the PacBio approach, or the Oxford Nanopore Technologies library preparation. And as Szi Kay has shown previously, by doing these two approaches, the library preparation in a similar way, you can compare pipelines.
So actually the bioinformatics pipeline between the two platforms are quite similar. So once you have your raw reads from PacBio or Iso-Seq, you go through a series of steps of processing and filtering. So for example, here you have your reads and you would remove any barcodes or primers that you've added during the library prep. And then you would orientate them from the 5' to 3' end. You would then align, so here you would we use Minimap 2 to align your reads to your reference genome. So in the case of human, HT38 with mouse NM10, and any lowly mapped reads are removed. Once you've aligned your reads, we then do a series of collapse and quantification. So for example, here we have the reads bucketed into based on splice functions. So for example, here we have these 3 reads that are collapsed to 1. These 2 reads here collapsed to 2, and so forth. So here now we're collapsing these reads into unique transcripts, which we then annotate using Squanty3. So here we now know what genes are present and where these isoforms are known. And we'll go further. But one of the really interesting things about now looking at a transcriptome is performing differential analysis. So previously in short-read RNA sequencing, because of these very short reads, analysis was typically restricted to looking at a gene level. So for example, here we have 2 conditions. We might be interested in looking whether a gene A, in this case, is upregulated. But the power of long-read sequencing relies on the fact that now we can look at the transcript level. So these are kind of multiple analyses at the transcript level, but what we're really looking at now is you can see expression changes between transcripts. So for example, in panel B, here we can see basically 2 isoforms which are both downregulated in condition 1 and condition 2, but by the same proportion. So here it's halved. What's really interesting is also we can look at scenarios where it's changed, but by different proportions. So for example, here again, it's downregulated, but by relative proportions. You can also have a scenario where it's changed, but the isoform, the dominant isoform changes between conditions. So here, for example, in condition 1, the orange isoform is the dominant isoform, but in condition 2, the upregulation of the blue isoform means that now the blue isoform B, is the dominant isoform. And the final scenario is where you have a change in the expression of the transcripts, but there's basically no gene expression change. So for example, you have a decrease in condition in condition 1 between upregulation of the red isoform by 3, but a downregulation of the blue isoform. So basically a net 0 change at the gene expression level. So all this is basically abundance, which in the past has relied on RNA-seq reads. So with gene expression or short read analysis, this is basically done by aligning short reads to a reference genome. However, now with the emergence of long reads, we can really rely on the power of long reads for annotation and do a hybrid approach where you align the short reads to your long reads, or in cases where the sequencing depth is very high, as Rosie will show later, you can then just rely on the long reads for both annotation and expression. And so I now draw your attention to a recent paper that Erin and I published in last year on Cell Reports. And what we found here was by sequencing the human and mouse cortex, we find widespread transcript diversity. So we found many novel transcripts that were not existing in previous annotations. And when we looked at the human cortex and the mouse cortex, we found some genes that were very different in terms of isoform number. I'll show you this later. And some of these isoforms were relating to genes of particular interest, such as autism or schizophrenia. For example, this gene here, MEG3, which is one of the most isoformic genes. So this is a UCSC genome browser track. Each line is an isoform. The colors represent whether it's novel or known. Things that are known are in blue. Things that are novel are in orange. So we can see just the novelty, how much novel transcripts we're picking up. Things here encountered is what's known, and again, we're already picking up isoforms that have very different splicing events across the gene. We did a whole transcriptome profile approach across human and mouse cortex. This QR code links to a database with all, if you want to type in your gene of interest, I'll show quickly what isoforms we're picking up. And what's really interesting now is because we are basically sequencing at a depth, we can now look at alternative splicing events. So as we go back, remember back to the previous slide, normally with constitutive splicing, you have your exon more present. With alternative splicing, you can get basically loads of different types of splicing events. So you can have, for example, an exon skipped. In this case, the orange is removed. You can have mutually exclusive, but it's either one or all. You can have an intron retention, where basically the intron is kept. You can have alternative 5 and 3' splice sites, where you can have basically different first and last exon. And what we find in our cortical dataset is a widespread range of splicing events across multiple genes, predominantly alternative first, but also exon skipping, which is in line with literature. And this is just an example of those events in a gene known in autism MEG3. So here we get again those exon skipping events, those intron retention, alternative first, but we're also picking up basically fusion reads. So this is a read which essentially reads through this gene MEG3 and DGBT3. These fusion read-throughs are very rare, but we're actually picking about 1% in our data set, suggesting they might be naturally occurring. And finally, we wanted to validate these novel isoforms. And to do this, we used publicly mass spectrometry data. So this is just an example where essentially we found a peptide that's highlighted in blue. So this is actually the final line. That span across 2 exons, suggesting validating essentially this inclusion of this novel exon, which is not existing in the reference genome. Using mass spectrometry data, we're already seeing the validity of these novel isoforms.
So we've recently been building upon all this work that's previously done by Szi Kay and Erin, and increasing our capabilities and number of samples to create long-read transcriptome datasets. And so here we're going to give you an overview of those. We have human cortex and the AD mouse models, and then whole transcriptome sequencing and targeted sequencing for these both respectively. So I'm going to begin by describing our human dataset, which consists of 47 prefrontal cortex samples. These range in ages from 6 weeks post-conception all the way up to 95 years old. This data was all performed on the Oxford Nanopore Promethean platform. So first of all, this just emphasizes the volume of data that we get from Oxford Nanopore long-read sequencing. So each line is showing a transcript, and you can see by eye, the high number, this was 189 different transcripts for the CUL1 gene. And you can also see visually that there's diversity between those different transcripts as well. So this is a bar chart of the top most isomorphic genes that we identified in our data set. The gene NDRG2 has over 2,000, and that's the most isomorphic gene. And you can see that the different colors correspond to the different, whether or not the isoforms are known or novel. So you can see by eye that the majority of these isoforms detected are in fact novel because they correspond to the green and yellow. But let's look at some genes that are more relevant to neuropsychiatric disease. So this is the top 10 schema genes. So schema is Schizophrenia Exome Meta-Analysis Consortium, and the top 10 schema genes contain ultra-rare variants which confer a substantial risk of schizophrenia. And you can see in this bar chart for the top 10 schema genes that the number of isoforms do vary that we detect in our data. But on the right-hand side, you can see that the percentage of novel isoforms detected is relatively high for all schema genes. Let's look at more detail at a particular gene. This is the EMX1 gene, which is a gene identified as a genetic risk variant in the PGC, which is the Psychiatrics Genomics Consortium, and that's as associated with schizophrenia. So as described before, these are tracks from the UCSC genome browser. Each line corresponds to a transcript, and the blue track at the bottom is existing GenCode transcript annotation. And you can see that I've highlighted in this red box on the left-hand side an example of the alternative splicing event that Szi Kay described, which is an alternative first exon. And then here in the middle red box, an example of alternative last exon. And then on the right-hand side here, we see two examples of novel exons which don't appear in the existing GenCode transcript annotation. Now, this data is from our whole data set spanning all ages, but we're interested in separating that out into postnatal and prenatal samples. So here we have postnatal only samples. There are 3 transcripts here which occur only in the postnatal samples. And then below it, we have transcripts which occur only in the prenatal samples. Now, the colors correspond to coding or non-coding status. And this is predicted using ORF predictions from the coding potential assessment tool. And so you can see the green corresponds to, it's predicted to be coding, and the red is predicted to be non-coding. And the other thing to note is that the shading of the color, the darker the shading, the more abundant the isoform is. So these are all quite pale in color. So these transcripts occurring in postnatal or prenatal only are actually quite rare. But what happened to the other transcripts? Well, they're shared between prenatal and postnatal. So here we've got the postnatal only and prenatal only at the top. And then here in the middle is prenatal and postnatal transcripts are shared, but shaded for postnatal abundance. And then at the bottom half, we've got prenatal and postnatal shared, but the prenatal abundance is shaded. So let me draw your attention to this transcript here in the middle. So this transcript, you can see that it's a much darker green. It's much more highly abundant. So and it's abundant in the postnatal sample. But this transcript is shared, so it also occurs down here, which it's also shaded dark green. so which indicates that it's also highly abundant in prenatal samples. So this transcript basically is appearing in both prenatal and postnatal samples at a high abundance level. But then let's also look at these transcripts which are shared, but are much darker green in the prenatal. So this indicates that these transcripts, although shared between prenatal and postnatal samples, they're much more abundant in prenatal samples. We can also look at the differential transcript expression between prenatal and postnatal cortex. And this reveals that there is a significant number of transcripts which have higher prenatal expression versus transcripts which have higher postnatal expression. Let's look at one of these genes in more detail. This is the CUL1 gene, which is again in the top 10 schema gene list. You can see that these two transcripts on the left-hand side, they have significant differences in isoform expression between prenatal and postnatal samples. So the isoform expression is significantly higher in prenatal samples in red compared to the postnatal samples in grey. And here's an example of differential transcript usage, which Szi Kay described at the beginning. This is for the VCAN gene. Here on the left-hand side, you can see for the transcript, in grey is the prenatal samples, and there's a higher percentage of isoforms which occur in the prenatal samples compared to the postnatal samples in red. And that's the same trend for this transcript here and along. However, when we look at this novel transcript that we've identified, this isoform usage switches so that the higher fraction of isoform is occurring in the postnatal samples. So not only can we look at the differential transcript expression between prenatal and postnatal samples, but we're also interested in comparing sex differences. And this is, of course, relevant for our study of neurodevelopmental disorders and neuropsychiatric disease in general. So you can see here that we looked in the HHATL gene, which is on chromosome 3. And for one transcript in particular, this one here on the right-hand side, you can see that there's a significant difference in isoform count between prenatal and postnatal samples. However, not only that, within the postnatal samples, you can see a significant difference in isoform count between male and female. So everything I've shown you up until now has been whole transcriptome sequencing. That's unbiased transcriptome sequencing of our samples. However, we can perform targeted sequencing. So we've taken a subset of the whole transcriptome sequencing samples, same age range, and perform targeted sequencing again on the Oxford Nanopore Promethean platform. So targeted sequencing involves using a panel of probes which are added to your cDNA, and they enrich for your genes of interest. So in a hybridization manner. So we selected a list of genes, 314 neurodevelopmental and neurodegenerative genes of interest, and designed probes, at least one per exon, for our genes of interest, which resulted in over 6,000 probes. And this panel of probes is added to our cDNA and then enriches for our genes of interest. And we then proceed with long-read sequencing. And the result is incredibly ultra deep targeted transcript sequencing data. And so here's an example of the kind of depth that we're looking at. So on the left-hand side, we have whole transcriptome sequencing. This is showing the gene GRIN1, which is associated with schizophrenia and autism spectrum disorder. We see 84 isoforms detected. And on the right-hand side, you won't be able to see any detail, I'm afraid. So that's because we see over 1,300 isoforms for our targeted transcriptome sequencing, and this is for the same gene, GRIN1. So we've also been expanding our data sets, looking at differential transcript expression between cortex and pancreas. So not only do we have targeted transcriptome sequencing data for the cortex, we also have targeted sequencing data for the pancreas. And this pancreas work was carried out by Ilsa, who's a PhD student in John's group. And we're beginning now to pick out differences in alternative splicing events and the diversity between cortex and pancreas, and that's ongoing.
Now switch gear from the human samples to something that I've been working on in my PhD, which is on a mouse model of Alzheimer's. So using PacBio, I also looked at kind of a global transcriptome profiling of a mouse model of Alzheimer's. And this really builds on the work from Isabel and her group, who published looking at 2 mouse models of Alzheimer's. For those of you who don't know, Alzheimer's has 2 well-known hallmarks. One is a plaque in the brain, and the other is a tangle. And we were looking into 2 mouse models that basically develop these respective hallmarks across a age. I'm interested in the TG4510, which expresses the human tau transgene. So at later time points, it basically develops these tangles. And what we found from her study was that looking at short-read RNA sequencing, we found a widespread transcriptional dysregulation, particularly in the immune response genes. So I want to take this one step further and sequence the same samples, but use PacBio long-read sequencing. And what I find is that at this level, we are now basically able to recapitulate some of those gene expression changes. So panel A is basically if I just use her short read RNA sequencing data and align it to my long read, we're basically seeing an increase in expression of GFAP, which is quite well known. So GFAP is an astrocyte marker that's upregulated in AD. But what's really interesting is that in panel B is that if I just use my long read counts, we're also seeing that upregulation at the gene level. The power of long read sequencing isn't just about gene expression, it's that actually now we can look at isoforms. So here, looking at GFAP, we're now being able to identify many novel isoforms. And what's really interesting is that if we look at the differential transcript expression, so now just looking at changes at the transcript, we see an upregulation of specific isoforms that appears to be driving the expression in at the gene level. So panel D is if we align the short reads to the long reads and perform at the transcript level. And panel E is if we just look at the long reads alone. What we both see is an upregulation of this blue isoform here. So now we're finding evidence that this increase in gene expression is actually predominantly driven by a single isoform. Some of you might notice that there's a few grayed out isoforms in the transgenic mice in the RNA-seq. And what we suspect is that when you start getting a lot of long read isoforms, when you align your short read to the long read isoforms, the algorithms get a bit not confused, but kind of complicates the alignment of these short reads. So it really just highlights the problems or challenges with short reads because you're starting to get these kind of ambiguous alignment. But with the long read, we can clearly see that there's only an increase in that one isoform. So having looked at kind of like a global expression change, I want to then kind of do a more targeted approach, because one of the issues with global transcriptome profiling is still the sequencing depth. So for this, we sequenced more samples across multiple ages, but using both PacBio and Oxford Nanopore. And for this, we chose 20 ADRS genes for IDT enrichment. So kind of in a similar manner to what Rose explained, we used IDT probes across these genes, which some of them are from GWAS, some of them are kind of from EWAS, and with this, we wanted to look at the isoform diversity of these ADRS genes in our mouse model. So this is an example of BIN1, which is an adaptive protein involved in endocytosis, but is also robustly identified in GWAS as an ADRS gene. And what we find is basically, again, a lot of novel isoforms. And to really capture the complexity, we've been, the heat map shows essentially where the complexity of these isoforms. So across the x-axis are the exons, and for each line, that's an isoform. And things in green are basically present exons, things in red are exons that skipped. And what we're seeing now is we can very clearly see kind of enrichment of exon skipping events across certain regions of the gene. So this is documented in panel C and clearly seen in panel B, where we have skipping events, but they seem to be particularly enriched or dominated in a region of the gene, possibly between exons 14 and 16. The same region is also dominated by intron retention events. So again here, I've just boxed here where between 14 and 16, where we're now seeing multiple isoforms with intron retention. And this region is in the collapse domain, which is particularly involved in the endocytosis. So we're now with a very thorough, ultra deep sequencing is picking up isoforms and really picking up splicing events that may be involved in have biological consequences. And what was really interesting with BIN1 was that when we looked between wild type and transgenic mice, we observed robust upregulation of two known isoforms. So here are the wild type mice, again 2468, and the transgenic. This is now just Oxford Nanopore normalized counts. We see a drastic upregulation of this red isoform and this blue isoform. And these two isoforms are actually structurally quite similar, with again multiple skipping events between exons 14 to 16. But one area of interest that I wanted to point out is in that exon 16 is skipped in the red isoform, but not in the blue isoform. We will come back later. But when we look at now isoform fraction, kind of the usage, this upregulation of this red isoform is always been dominant across the months between 2 to 8 and then wild type and transgenic. But what's really interesting now is with this upregulation of this blue isoform, we now also see it becoming more dominant in the aged transgenic mice. And finally, if we look at the expression level, there is actually no change at the gene level, suggesting that while we have pick up these kind of changes at the transcript level, they seem to be a normalized, or again, net zero change at the gene level, highlighting the importance of doing novel sequencing at the transcript level. Finally, I just wanted to point to my final slide, which is basically an integration, a kind of an omics approach. So John's group is not only interested in splicing, but also epigenetics. And with this data, in the same samples, we also had methylation data. So one thing that I was really interested in looking at was to integrate the two. And in BIN1, what we found is a differentially methylated position in the same mouse model. So here across time points again, 2, 4, 6, 8, wild type and transgenic, we see an increased methylation in the transgenic compared to the wild type at 8 months. And what's really interesting is that the MP is actually located upstream of that exon 16 that is skipped in the red isoform, but not in the blue isoform. Which is really interesting because it suggests that maybe there might be some interaction of splicing and epigenetics. So moving forward, we want to first validate the expression and the methylation, but also then to use CRISPR to see if there's actually a functional relationship between this methylation and the splicing at this point. With that, I would like to conclude our presentation and to acknowledge our funders and also to John Moore. So have any questions? Oh yeah, happy to.